Good morning, my colleagues at the International Society for Computational Biology Conference. My name is Adeoti Olatunde Michael from Landmark University. I am here today to talk on the computational screening of promising epitopes from Mycobacterium tuberculosis ESS genes for a multi-epitope vaccine design. The key concepts of this topic include computational screening, that is the use of computational algorithm for to analyze and model large sets and predict certain biological outcomes. Promising epitopes. Epitopes are small segments of antigen that are recognizable by the immune system, specifically T and B cells. A good vaccine should be able to trigger both humoral humoral immune system as well as cellular immune response. The ESS genes are systems of specialized families of bacteria which include Mycobacterium tuberculosis, which are basically responsible for the proteins of certain molecules on bacterial membrane and is actually very responsible for the virulence of bacteria in which they use in immune modulation and cellular communication. Also, multi-epitope vaccine is going to be the one that we incorporate different multiple epitopes which are pathogen based into a single vaccine. Mycobacterium tuberculosis are now recognized to be the human pathogens, which is made up of more than 50 species of human pathogens. Mycobacterium tuberculosis highly aerobic, which requires a high level of oxygen and is primarily responsible to infect the respiratory system of human. Mycobacterium tuberculosis, which was discovered in 1882 by Robert Koch. Uh, Mycobacterium is also recognized to be metabolically versatile because of their ability to be able to withstand certain stress by switching their pathway when under stress. It is known that in 2020, more than 1.7 million died of TB, while 1.8 million died of COVID, making TB to be responsible as the second largest killer. And it is believed that TB kills more than 4,000 and infects more than 30,000 people daily, according to the WHO. TB prevention has been since 1920 has been basically by the use of BCG vaccine, which is primarily used in childhood immunization program. Bill Gates in the year 2010 actually stated that this decade should be a decade of rational vaccine. That actually opened the door for uh, research to be towards developing TB vaccine. And don't forget that BCG only protects newborns and infants. Why are we interested in multi-epitope vaccine? Just like I said, that a good vaccine and an ideal vaccine should be able to induce both CD cell as well as a CD4 and CD8. And multi-epitope vaccine is going to uh, we're going to address these above issues, which is compared to the traditional vaccine, which has a very low antigenicity. The aim of this study is in line with Goal 3 of the Sustainable Development Goals of Goal 3, which is good health and well-being, and it is designed to design an effective multi-epitope vaccine by the use of a computational method. This is the reference genome of H37RV, which has a genome of more than 4.4 million base pairs. In this study, a retriever of the Mycobacterium tuberculosis was performed by the use of H37RV as the reference genome, and this was retrieved from Uniprot's database in FASTA canonica, and this will aim in order to be able to actually do. So after that, the retrieve sequences, the antigenicity was performed, as well as homology, whether it has the probability to be able to develop the same, to react with the gene of human, and the subcellular localization was also performed. Also, gene essentiality was performed, and after retrieval of this, the epitope prediction process started, and the linear B cell epitope was predicted by the use of BC Pred, baby Pred, ABC Pred server, where after which the toxigenicity and antigenicity and immunogenicity was performed by the use of toxin Pred, Faxigen, Epitopes, and IDP using an in silico method. And at the end of the day, 19 epitope B cells was gotten. Then the next one was to predict the cytotoxic CTL epitope sequences, which was done by the use of IDB server. Toxin Pred was used for toxigenicity prediction, allergenicity, and antigenicity. At the end of the day, a 49 CTL, that is cytotoxic cell unique epitopes, were identified, after which the HTL lymphocyte epitopes was also performed and 11 HTL epitopes was retrieved, and the sequence is as predicted, displayed on the board here. So summarily, the computational workflow I use for the design of this multi-epitope vaccine includes retrieval of core proteome, followed by the antigenicity and localization or location prediction was performed by the use of Faxigen, Cello, and Cello server. After which prediction and evaluation of cytotoxic lymphocyte epitopes was performed by the use of IDP, Epitope, Toxipred, and Faxigen, after which it was subjected to the prediction of an assessment of helper T cells by the use of IEDP, Epitope, Toxipred, Faxigen, and their capability or ability to be able to induce interferon, interleukin 4, as well as interleukin 10. After which the prediction and assessment of linear beam lymphocyte was used with the use of ABC Pred, Bepi Pred, and Toxin Pred for toxicity evaluation. Then after which there was a performance of estimation of population coverage by the use of IEDP population coverage tool, and a vaccine construct was performed. That is, a multi-epitope vaccine was performed. The sequences will be displayed in the next slide. After which this was subjected to the structure prediction and analysis by the use of PSR Pred, Faxigen, IDP immunogenicity, and Galaxy refined server. Docking was performed by the between the prefaxing protein, that is the MEF vaccine candidate. The toll-like receptor 4 was also used as the ligand for to dock the vaccine candidate. After which, this was also simulated by the use of molecular dynamic simulation, and the immune simulation was performed by CIMM SIM 10.1 server, after which it was codon optimization was performed by the use of a GCAT server. Just like I said, the vaccine candidate is as predicted here, which are variously linked with different linkers as displayed. This was subjected to physical chemical parameter testing by the use of protoparam server, and the number of it was discovered that my vaccine was suitable with the number of amino acids to be 1,344 with average molecular weight, and the theoretical PI is basic, which is around 9.7 as display here, and it was predicted to be highly antigenic with no allergenicity reported, and very soluble, moderately soluble. And this is the structure of the predicted secondary structure of the multi-epitope vaccine as predicted. And this one is the result of the secondary prediction by the use of PSI PRED of multi-epitope vaccine protein, which was predicted. And this is the result of the homology modeling by the use of Ramachandran plots, and which actually allowed the region to be 95.8 to be within the allowable region, and the three-dimensional structure, that is the tertiary dimensional structure, was performed as displayed by the board. Then the disulfide engineering was also performed as 8WCT was the similar protein as was retrieved from protein database, and this is the result of the interaction between the ligand, which is TLR4, that is, and the predicted vaccine. The molecular docking was performed by the use of IMOD server as being displayed. Just like I said, the codon optimization was performed, and the green side is where the predicted multi-epitope vaccine was inserted, and the immune simulation was performed by as displayed, and it was relatively found to be antigenic, and will be able to trigger good immune response with the host, just like this. And the immune simulation was also performed by the use of active memory, which triggered the cell proliferation according to when it is exposed to multi-epitope vaccine. Summarily, the adjuvants used in this study, the predicted epitope were found to be higher with very low antigenic scores, and reliably they can be used universally as predicted for my vaccine candidate. Also, the physical parameters suggested stability of the predicted vaccine in formulation, manufacturing, and also in the storage. Also, the residue of non-polar, polar, and hydrophobic aromaticity suggested that the predicted vaccine is also going to be stable chemically. Also, the secondary structure provide an insight for us to be able to predict the functional properties in terms of the immunogenicity and ability to be able to interact with the immune system. Also, the 3D structure, as predicted here, also gave us an insight that also help us to be able to identify the binding site for the antibody stroke T cell. The refinement has been performed by Galaxy show that the conformational stability and his ability to elicit immune response is very, very positive. And the disulfide engineering also suggested the stability in the tertiary structure, which maintained the conformational stability of the vaccine candidate. The Z scores, according to the Ramachandran's value, suggested a most favored region of the predicted multi-epitope vaccine to be within the 95.8% as shown in the displayed in figure 3. Also, the ligand also showed that there is going to be pattern recognition in terms of innate, which is also leading to robust immune response. Also, the codon optimized multi-epitope vaccine construct suggested its ability to be cloned and expressed in E. coli, which is actually used for in vivo study. Lastly, the immune simulated plots suggested a prolonged memory due to if the predicted vaccine actually taken. And these are the list of the selected references that actually guided this study. Thank you for your attention. Thank you.